Thank you. First of all, thanks to Carmen and to all the organizers for inviting me here. It's really a great meeting. I have so many new ideas already. Yeah, I'm really happy to be here at this conference. Also a little bit embarrassed because most of what I'm going to be showing you today is not about ME/CFS, but about long COVID. But I can assure you, the people here, people online, that everything I will show today is something that we will also study in the context of ME/CFS. So, I was asked to talk about autoimmunity in long COVID today, our work. I think when I talk about long COVID, particularly for this study, autoimmunity, it is important to make a general distinction between the two groups of long COVID patients. One that had initially a severe disease, got hospitalized, inflammation, hyperinflammation, tissue damage, and the group that did not have initial severe disease but mild disease, were non-hospitalized. First group contains more males and the second group more females. What I will be focusing on today is the group that initially had a mild disease. They're both important to study, but I will focus on this group today. So we've heard it before about long COVID, ME/CFS. There's different hypotheses for long COVID and ME/CFS, and we're in my lab actually studying multiple of these hypotheses. What I will focus on today is autoimmunity, but I would like to state clearly that I do not think this is like a single mechanism going on in the pathophysiology. I actually think that autoimmunity is linked to these different other hypotheses as well. We will hear other talks about molecular mimicry, by which you can link autoimmunity to viral infection, for example. Microclots are known to be full of IgG antibodies, and I will try to make myself a link to metabolism and autoimmunity as well. So we wanted to study autoimmunity. Why? Well, already pretty soon after the pandemic, and when these long COVID patients started basically dripping into the hospitals, it was clear that both COVID-19 but also long COVID patients express many autoantibodies, but the big question is, are they actually contributing to disease? Are they actually causing disease? So it's correlation versus causation. And that's something that we wanted to address. And we did that actually in a similar way as David Petrino already talked about this morning. What we did is we took the blood of healthy control and long COVID patients. We figured if there's autoimmunity there playing a role, we can take out the antibodies, and we focused on IgG. IgA may actually be also an interesting one, but I'll talk about that next year perhaps. We transferred those to mice to see if they would get symptoms. And we did that by reading out with a whole panel of behavioral tests. Now, we started doing this a little bit before Akiko Iwasaki and David Petrino did this, but we were definitely not the first doing this, because it has been done before in the disease fibromyalgia. Great work from Swenson and the Anderson group, showing that you can transfer these antibodies and you get symptoms in mice as well. So that's what we wanted to do. And we thought it was an interesting thing to do as well. We wanted to plan it, and then people said heterogeneity. There's so many, the heterogeneity in this group is so big, you need to start working on subgroups. And that was actually a pretty good idea. But where do you start? So we start where most people start, and that's just how are, in blood markers, how are patients different from healthy control. So here we compare in green the long COVID patients, in white the healthy controls. We started looking at all these different proteins. We didn't find any differences in the pro-inflammatory cytokines in this group because they were initially mild disease. If you look at the group initially with severe disease, then you do see those pro-inflammatory cytokines popping up. But we did see differences in the top panel here in the type 1 interferons, and the bottom panel in the neuronal damage markers. So we thought, okay, there's a few patients there with elevated levels. Let's see if we can group the patients based on this serum phenotype. And that's what we did. In the end, we ended up with 3 groups, which we color-coded: red, gray, and yellow. In the end, the red group showed most pronounced increases in the neuronal damage markers, indicated here on top. The gray group showed increased type 1 interferon production, and the yellow group actually had no elevated markers compared to the healthy controls. And of course, we wanted to dive into this a little bit deeper, so then you go to the serum proteomics, O-link, really great way to burn a lot of money. But it seemed to make sense. We had the red group popping up with neuronal damage markers mostly, the gray group with muscle markers, and the yellow group with immune cell markers. Clearly 3 groups, and then we could actually start doing these experiments. So we purified IgG from about 10 to 12 patients per group, injected them intraperitoneally into these mice, and we did a whole battery of behavioral tests, and I'm not going to show them all to you. Many of them actually didn't show anything, but some of them did, and those were the most interesting ones, of course. So one of the things we did was just monitor in an open field test to see after injection how much do the mice walk, and how fast do they walk, and how long are they standing still. And that gave some interesting phenotype as well. So here on the x-axis is depicted the time, on the y-axis the distance that they're walking. In black, the black line, the dashed black line is healthy control, so mice injected with control IgG. That's the baseline, and as you can see, the yellow group and the red group actually show no phenotype, but the gray group does show something on day 1, where it's significantly impaired, both in their how much they walk, but also how fast they walk. But we did many more assays, and the most striking example we saw with the pain tests. And one of the pain tests was a von Frey test, in which you gently poke the paw. The mouse is standing on a grid, and you gently poke the paw of the mouse, and if they're in pain, they start to retract with very little activation, already start to retract their paw. And what we saw there was this. So again, x-axis the time, y-axis is the mechanical sensitivity here. So the lower is more pain. In black is indicated the controls again, and as you can see, all group had a very severe phenotype here. Interestingly, the gray group, which showed the phenotype, the only group showing the phenotype in the movement, had the least pronounced phenotype here, but particularly the yellow group right away had a severe phenotype. This is a log scale, so this is really severe pain experienced by the mice. The red group was somewhere in between, initially basically following the gray group, but then at day 3 really the phenotype kicks in and it enters the level of the yellow group as well. So we really had a clear phenotype just by transferring the antibodies, the IgG antibodies from patients to mice. And for us that, it really showed that there was a causative role for these antibodies. Just transferring them can actually cause symptoms. But on top of that, it was really interesting to see that we also seem to be able to transfer specific symptoms from patient groups to mice, the differences between the gray and the yellow group, for example. So yes, this is an indication for a causative role of autoimmunity in this disease. And we're now really busy focusing on other related diseases, post-acute infection syndromes. And we're doing this now already for ME/CFS. We want to set this up for post-treatment Lyme disease syndrome as well, and in the future we would like to do this for Q fever fatigue syndrome too, and perhaps even other related conditions. We submitted our preprint about this about a year ago or so, but as you know these days peer review takes forever, so thank God for preprints. But the experiments that we need to do actually did help us a lot, and what we did, our experiments had been done a long time ago already. So we took from our patients, long COVID patients that we initially used, which are now already like 2 years ago, the patients that still suffered from symptoms, we drew blood again, purified the IgG, did the same experiment, and the phenotype was still there. So that's really a nice validation. But on top of that, of course, what Dave Petrino already showed was a few weeks after our preprint, they also posted their preprint, basically showing the same thing, that IgG transfer induces symptoms. And there was even a group presenting a poster or an abstract at a conference, a group from Belgium showing the same thing. And I think that's really important, that this is, that these findings are validated by different scientists in a different country using different patients, indicates that it's really robust and is, hopefully real. Of course, he wanted to know, what are these autoantibodies directed against? We don't know. So we did a screening, and the largest screening that we could find was a screening by CDI laboratories, at least commercially available. 21,000 autoantigens, which is quite a lot, but probably doesn't cover everything still. What we did see here, when you look at the 3 groups again, so the red, the gray, and the yellow again, they do have distinct autoantibody profiles there. So it really seems to make sense that there's different antibodies going on there. And it's just not just one magic bullet, but it's a whole array, and patients actually have a different different collection of these autoantibodies, while healthies in this case do not. We also wanted to zoom in a little bit and go to the individual patients, and then you basically see what we all know, that every patient's different. Everyone has their own set of autoantibodies, but we wanted to find the common denominator here. So we started looking into what are these, what are the proteins, where are they expressed, the proteins that these autoantibodies are directed against. And what was interesting is that many of these proteins are actually expressed in the central nervous system, in the peripheral nervous system, or is expressed in skeletal muscle. So we wanted to do a little bit of follow-up on that. And one of the things we did was just take brain slices from mice, which we, of course, still had from our experiments as well. But if you, here on the left, if you incubate these brain slices with IgG from a healthy individual or a recovered individual, so got COVID-19, did not develop long COVID, we never see staining. But if you do that for a number of long COVID patients, and I think about 20%, we do see autoreactivity. We see these autoantibodies binding to neuronal structures. And particularly in this case that I show you here, really specific regions of the brain start to light up, such in this case the cerebellum and the hippocampus, which, well, according to neurologists that I talk to, say, that could be related to the symptoms that you see in long COVID patients, for example, the brain fog, the memory, and stuff like that. That's basically the central nervous system. But we also wanted to try to see if we can link our autoimmune work to the work on muscle abnormalities that Rob Wust showed, and has been mentioned many times already at this conference, to see if we could find a link. Now, Rob indicated many muscle abnormalities, after exercise of these patients. But what he also found was focal necrosis, really skeletal muscle cells dying, and that was interesting to us. So together with Rob, we did experiments just culturing. This is still very much in its infancy, but we started culturing these skeletal muscle cells, and we exposed them to, for example, a plasma of healthy individuals, and they just grow happily in monolayer. But what you see for some patients that I selected here, when you add the plasma of these patients, these skeletal muscle cells actually start to die. And sometimes it's subtle, sometimes it's extremely pronounced, and basically 80% of the cells die right away. Now, this is still very early start of this, and we want to follow this up with actually culturing skeletal muscle cells, letting them exercise, and creating PAM in a dish. But perhaps, part of this whole equation is autoantibodies and the exercise, and that's something that we may study in this way. So where do we go from here? We have basically 3 ideas with the autoimmunity work that we want to do. First of all, basically what we have done is create an in vivo model for long COVID, which is nice, which you can use to test therapies, for example, but it is dependent on the blood of patients, and we want to get rid of that. So what we're currently actually doing, or have already done, is to isolate the autoreactive B cells, to sequence them, and then we can produce these autoantibodies as much as we want. And we can actually basically also hand it out all over, so everyone can actually start these specific in vivo models, and even perhaps a specific subtype that you want. You want a red long COVID mouse, or a gray one, or a yellow one. Of course, it gets way more complicated than that. Probably it is a combination, but that's something in the future we want to go to. Another thing is the in vitro models. Yes, you can test therapies in an in vivo model, but it probably would be way more efficient. And you could do it on a way larger scale if you could do drug screening in vitro. So we're currently setting up models for skeletal muscle, cardiac muscle, and neurons, culturing in vitro, exposing them to either the plasma and the antibodies that we get from patients or our recombinant antibodies, to see if we hit a phenotype. And if you hit a phenotype, you can actually test drugs. And that can be done in like a personalized way. You just take antibodies from one individual, you hit a specific phenotype, and you can see for this patient that may work. Again, far in the future, but this is where we want to go. And third is the diagnostic test. So we have identified several autoantibodies that we never see in healthy controls, but we do see in long COVID patients. And based on that, we actually have currently set up a Luminex panel of about 20-ish autoantibodies that we want to use to basically test whether we can use it as a diagnostic test. A bit comparable to what we currently have for myositis, which you apparently also have like 16 different autoantibodies, which indicates that you have different subset which require different treatment and have different risks, for example. That would be great to have for long COVID and ME CFS, of course, as well. Now, on top of that, we're also starting 3 clinical trials, randomized clinical trials, this year in Amsterdam. I'm looking forward to number one, that's immune adsorption. That has been done, of course, also in Germany a lot before. Of course, immune adsorption, taking out the IgG antibodies, including the autoreactive one, will only help if patients are really autoimmune. So we want to do this, but we want to, as an inclusion criterion, we want to base this on our autoantibody profile that we see in these patients to see if that helps. I added a QR code for a movie. I will also show it in my next slide. But we have 2 other clinical trials going on as well, and I think it's all interconnected. The other one is, number 2 is about sonlicromanol, which is a drug that can help boost the mitochondrial activity. And the third one is an inhibitor for the enzyme IDO2, which is, well, in many cases, strongly activated in at least long COVID. Well, that may also help with the whole tryptophan metabolism, etc. And that is basically what I wanted to tell you today. This is the paper. I'd like to thank all the authors involved, particularly the primary authors indicated here in bold. Thanks, Carmen, again for the invitation and all the other organizers to present here. I'd like to thank Risa Pretorius and Akiko Iwasaki because they're just awesome. I'd like to thank the funding agencies and, of course, all of you for listening. Thanks.
Thank you, Jeroen, for showing the pathogenicity of these antibodies. You didn't mention any test to evaluate new drugs, either in vitro or even in vivo.
Yes, I mean, that's ultimately where we would like to go to. I think we need to have the phenotype very solid first. The heterogeneity is very big. So I think we would need to have a very clear and reproducible readout first, either in vitro or in vivo, before we can start basically testing or screening these drugs. But that's definitely on our list as soon as the assay is robust.
Thank you. There is an interesting question.
Very interesting talk. One thing I want to say, I'm not an ME doctor, I'm an experimental immunologist, and you have to be aware that when you pure IgG, human IgG, and give this into mice, you do an immunization or vaccination, and you will have a very strong immune response to human immunoglobulins. So what are the controls, and have you done ordinary IgG from healthy humans as a control? And do you see some of the same effect?
No, it's a really good point. Just injecting something in a mouse, of course, will induce responses. And it does to a certain extent. But indeed, our controls contain IgG purified from healthy individuals. Initially, we did IgG pre-pandemic. Later on, we repeated this with IgG post-pandemic because who knows? You see a little bit of a phenotype there from the control IgG, but it's very limited. And the long COVID, all the long COVID samples really stand out. Of course, you will get autoantibodies from the mouse against human IgG, but it takes a while to develop, probably around a week or so. And we see the phenotype basically already popping up at day 1, 2, and 3. So I think that effect is rather limited. Yet the autoantibodies that we're basically sequencing and producing ourselves, we can of course provide a mouse backbone, so we won't have that problem.
Thank you very much. I think we proceed, but a lot of patients or people here in the internet have also thought, okay, now it's our autoantibodies, what is now the best? And I think it should be mentioned that the presence of the pathogenic autoantibodies is not an easy game to deplete those antibodies. We know it from autoimmune diseases where we use CAR T cells or a lot of B cell depleting drugs, so that they, for example, rituximab is not able to deplete all these pathogenic antibodies in the lymph nodes, in the B cells, and also obinutuzumab and others. It's very difficult, and even in CAR T cell therapy, which diminished all the B cells, but even there, these antibodies are present, and it is not easy to really access the antibodies. Maybe it's different in ME/CSF, but in autoimmune disease it's very difficult.
No, definitely, and of course you need to make a distinction between B cells and plasma cells. If you get rid of the B cells, the plasma cells still start to produce, of course. Yeah. So we have some interesting talks about that later, I think. But also, I do not think that every patient will have autoimmunity. So we had groups of patients that we injected of 10 to 12 patients. Theoretically, it could be one patient having the pathogenic antibodies and causing the phenotype, right? So we really, in the end, need to split it up into individual patients to see if we can do these experiments as well. But it would require a lot of blood and a lot of mice, of course. But yeah, so we do see a lot of autoantibodies in these patients, but if they're really pathogenic, that's something that we still need to investigate further, definitely.
Thank you very much for this clarification.